Hello everyone and welcome to today's event with Explain the State of Your Plants. I'm very excited to welcome this very special guest. It's a very trendy topic that I'm very excited about. So looking forward to sharing that with all of you. So we're gonna discuss CRISPR. It's an acronym that you might have never heard of, but today's the spot to discover what it means. So we have a very special guest with us today as part of the Gairdner Foundation series. So Rodolphe Barrangou is here with us today. I'm going to welcome him on stage very shortly. So you can say hi, and then we're going to go through a short presentation together, and then we're going to have a lot of time for your questions. So keep them in mind, write them in the chat, put them on paper if needed, but we're going to have a lot of time to discuss with him afterwards. So please prep your questions for Rodolphe that I'm welcoming on stage right now. Hi!
Hi, Alex, and good morning, everyone. It's a pleasure to be here. I'm indeed here to talk about CRISPR as a great example of how to explore by the seam of your pants. And I've had the great privilege to work on CRISPR for 20 years. And it's been a journey for sure, but it's only the end of the beginning. And I can guarantee you that there's a lot of aspects of your life that will be impacted by CRISPR, that are being impacted by CRISPR, maybe unbeknownst to you, have already impacted your life. So let's go with the show. Alex, if you don't mind, share the screen and I'll kick that off.
Awesome. See you in a few minutes.
All right. So as mentioned, I was a 2016 Gairdner International Award recipient, and part of the foundation's job is to celebrate great science, to convene great minds together. That's what we're doing here this morning, and to inspire the next generation of scientists and consumers, which includes largely the most of you here. So I'm going to talk about CRISPR in general as a technology to do genome editing, and discuss in the next 25 minutes the many applications and the many implications of this amazing technology. And most scientists in the world are somewhere on that spectrum. And the job that we have is to try to translate great science, great discoveries, great exploration, sometimes into useful technology. And then occasionally that technology can be applied practically to work on tangible products that will be endorsed and accepted by consumers, endorsed and accepted by regulators, endorsed and accepted by patients and students, of course, throughout the world, and then turn science into products that make the world better. This is hard. This is difficult. It takes a lot of resources, lots of people, lots of money, depending on the field. And oftentimes you should realize that whether you do science in academia, we do science in industry, we do science policy in the government, as a societal consumer and or patient and or investor and or student, we all have to do our part to turn that science into products. And in the case of CRISPR, I would argue that we're here right now. We're at the end of the beginning where some applications are turning into very exciting, very tangible products. And the speed at which the CRISPR science has turned into technology that's used and commercialized is amazing. So to give you some context of where we are right now in the CRISPR world, there's about 300,000 labs in the world that are using CRISPR all day, every day. And as a matter of fact, I'm in North Carolina. I'm a professor at NC State University, and we have high school students and even sometimes middle school students doing CRISPR experiments in the lab, in the classroom. So it's very explosive, and it's changed and grown quite a bit in the past 10 years. Of course, we're here primarily in the US and Canada today, but those CRISPR tools have been deployed and shared with the whole world. There's about 108 different countries that have schools and academic institutions that have received some CRISPR constructs to play with and tinker with and explore with. And it's very competitive because there's 50,000 people from 5,000 different universities that have published at least one CRISPR paper, and you can manuscript, like a scientific study. And you can see here that internationally, there's a lot of aspiration, there's a lot of activity, there's a lot of development, there's a lot of research, and there's a lot of commercialization that's happening primarily in the US, but also in China, many parts of Europe, and of course, Canada also in the top 10 here. The bulk of the work is occurring in human cells, in blue here, for human therapeutics, but there are parts of the world where CRISPR is being deployed to change DNA, not just in humans, but in crops, in livestock, and in bacteria used throughout biotech and the food supply chain. And there's now dozens of companies and startups primarily that are using this disruptive technology to change the world. So given the fact that this is where we are, how much and how fast is changing the world, let's try to go through how in just 10 years we've gone from science to product through those different steps. So as a matter of fact, CRISPR is an acronym, as Alex mentioned. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a very descriptive, very accurate, very cumbersome mouthful. And what CRISPR is in nature is a genetic iPhone. Right? You all have your phones, and you likely know you don't use your phone to call people. You don't use your phone to browse the internet. You use primarily a feature on your phone called a camera. And what CRISPR is in nature in many ways is a genetic camera that takes a snapshot of a piece of DNA from an invasive element like a virus, and then integrates that into the CRISPR locus to have sequence-specific targeting and cleavage. And what CRISPR is in nature is an immune system that takes and steals and copies and pastes a piece of viral DNA to have sequence-specific targeting and cleavage. This is why I got the Gairdner Prize back in 2016. We showed biologically that CRISPR's immune system in bacteria, and that this immune system specifically and selectively and accurately cuts and shreds viral DNA. Now, scientifically, it's cool, it's interesting, it's peculiar, it's idiosyncratic, it's mesmerizing. But the reason we're here today, the reason you've heard about CRISPR, is not because of CRISPR science, it's a CRISPR technology. And what my colleague Jennifer Doudna and Manuel Charpentier did a few years ago was win the Nobel Prize in 2020 for turning CRISPR, the immune system, into CRISPR, a technology. And what I told you about CRISPR in bacteria being a molecular scalpel to cut viral DNA can be reprogrammed and repurposed by scientists to just be a molecular scalpel to cut any DNA you want. Think of the power of Word and Control F, and look for a sequence in DNA and selectively use that molecular scalpel called CRISPR to cut the DNA there precisely. And what it's used for is not just cut DNA. When you cut DNA, you generate a mutation, and you have the ability to edit the DNA sequence precisely at the site of cleavage. That's what is called genome editing. We can cut the genome and rewrite the genome precisely at the site of cleavage. We can turn that G into an A. And if you look for a string of letters like Sir William Shakespeare's famous words, to be or not to be, that is the question, you could program CRISPR to look for that string of text in the DNA and cut between that and is, and edit the text in Shakespeare's words, so to speak, and say to be or not to be, that's the question. To be or not to be, that is the quintessential question that defines humanity, or to be or not to be, hashtag the question. We have the ability to rewrite DNA. Of course, in the past 10 years, there's a lot of genome editing technologies that have been developed, not just to rewrite DNA or change one letter, but change the whole world, change a whole chapter. People want to have the ability to change 100,000 letters in DNA. So you can take out a block of sequences, you can add a block of sequences, or we can engineer CRISPR to change punctuation, change transcription of DNA, turn it on, turn it off, turn it up, turn it down, or even change the epigenetic state of DNA, the mother's imprinting in children yet to be born. Now, in the past 10 years, those 300,000 labs and 50,000 authors throughout the world have been very hard at work turning a CRISPR into a molecular scalpel that can edit all kinds of genomes. Model organisms, as you see at the bottom here, viruses, bacteria, fish, model organisms like chimps and plants, all the way to food and agriculture, crops, obviously, livestock, obviously, think not crisp chicken, but CRISPR chicken, not crisp bacon, but CRISPR bacon, and the like. And then, of course, in medical applications, all kinds of organisms and cell types we can use to develop medicines. To illustrate the power of CRISPR here, take my word for it, the wing pattern color scheme of a butterfly is unique in the world, much like the human iris. No two butterflies have the same wing pattern. CRISPR is so powerful that a butterfly geneticist can take some cells out of this butterfly, program CRISPR to target a gene called yellow, which predictably is responsible for the coloring yellow in the wing patterns of butterflies. And we can grow another butterfly, as well as a bit smaller here, because it's younger, that has the exact same wing pattern in which the color yellow has been removed. and they otherwise have the exact same wing pattern, the exact same DNA sequence. Of course, you can do that with other colors. The point here is that CRISPR is so powerful, we can recolor the wing pattern of butterflies, suggesting that the only limitation is our imagination. And as a matter of fact, in the last 10 years, people have been using CRISPR to recolor the eyes of insects, as you see here, recolor fish, breed crops, change one or the other eye, make hypoallergenic cats, breed, of course, livestock, breed trees and forests and crops, and then develop human medicines that can address genetic disease. This technology, as a 10-year-old, is an amazing 10-year-old that is disrupting molecular biology, disrupting medicine, disruptive agriculture, disrupting the food supply chain, disrupting biotechnology, and changing the world, because we can virtually rewrite any DNA we want in any species we want. It's a technology that has many advantages, a few caveats, and as long as we know how to predict DNA repair and how to deliver it to patients and or plants and or bacteria and or viruses, we can change DNA at will. So how are we going to apply it? Well, there are multiple types of industries being upended, being revolutionized, being impacted, being explored, exploring by the seam of your pants, so to speak, today in biotechnology. So any bacterium, any yeast, any algae used for food fermentation, biomanufacturing, household care, think detergents to do laundry, bioenergy, think biofuel at the gas station, have had their genomes edited and enhanced using CRISPR. It's the same in agriculture, whether it's a crop, whether it's livestock, whether it's a food bacterium or yeast, whether it's a tree, a flower, an ornamental, aquaculture, fish, seafood, check, even grasses. All those organisms have had their genomes enhanced and or bred using CRISPR technology. And of course, in human therapeutics, you can correct genetic disease, you can target viruses, you can target bacteria, you can change the microbiome, you can engineer cells for cell therapies, immunotherapies, you can develop new tissues for human xenotransplantations across species, we can do gene drives, we can do that in animals, diagnostics, pet care, and so on and so forth. So this technology was developed just 10 years ago. How far and how fast have we gone just in the last 10 years? Well, as of this week, I just changed the numbers. There's already 80 different CRISPR clinical trials underway, dosing humans throughout the planet. And as you saw earlier today, now we're just talking about the clinic here. The whole world is doing CRISPR trials. Most continents doing CRISPR trials. There's already 8 clinical trials that have been completed, including sickle cell disease and cancer. There's 28 different CRISPR trials throughout the world that are actively recruiting patients, including 12 right now with interventional studies where they're trying to dose patients to cure their genetic diseases like beta thalassemia or sickle cell disease, or trying to change and address the cancer cells. And as a matter of fact, if you look up on Google Victoria Gray and CRISPR, you will see that 3 years ago, the first patient for sickle cell disease was treated one time with the CRISPR cell therapy, and she's been in remittance ever since. It's magical. Science fiction, but just the science part. And there's a lot of clinical trials underway, as mentioned, a lot of FDA engagement, as mentioned, and lots of companies with whole pipelines to develop new drugs that address TTR, sickle cell, beta thal, and other things, whereby we can deliver CRISPR with a guide into humans intravenously. So those molecular medicines would go into their liver, enter liver cells, inside of liver cells, enter the nucleus, inside the nucleus, change the DNA to correct genetic disease. And the same technology is being used the same way in organisms across the tree of life, like plants, like vegetables, like fruits, like trees, like livestock, like bacteria, like yeast, like mold, and others, to revolutionize the food supply chain. Leaner pigs, higher yield, rice, drought-resistant corn, pest-resistant cattle, non-browning mushrooms, or healthy, sustainable yogurt. As a matter of fact, yogurt fermentation was the first ever application in which bacteria were CRISPR enhanced to be phage resistant. And all the yogurt and cheese in the world that's not homemade has been CRISPR enhanced and commercialized since 2011. And we're using this right now to breed cattle, to make yogurt, to make cheese. In Japan, they have CRISPR tomatoes. In the UK, they have CRISPR tomatoes. And you can see how this technology is being used to build a resilient, a more sustainable, and much healthier food supply chain through innovation and technology. And as a matter of fact, you can see that ever since 2019, crops have been commercialized. And there's all kinds of organisms like maize, like tomato, like soy, like fish, the sea bream and pufferfish. I mentioned cattle in particular, pigs and chickens, and of course cattle, the three major livestock across the food supply chain in the US. And of course things like greens, mustard greens, pennycress, lettuce, I mentioned mushrooms and others, being deployed not just in the US, but increasingly so throughout the world, including in China, soon enough in Europe and part of South America. Now, with great technology, with great potential, with great excitement, with great scientists, with great science, and with great products, also come great expectations and responsibilities. And as you can imagine, in today's world, it's very important to ethically and responsibly and equitably use science and technology for humankind and for the planet. So ethically, we have to ensure that the perception of the public at a time of anti-science and skepticism narratives is perceptively agreeable and positively perceived. We have to go fast enough to save human lives and feed the world, but not go so fast that we make people uncomfortable. And we have to be equitable by providing access to those sometimes ill-affordable technologies to the whole world. We have to be responsible ethically. We have to make sure that the scientists and the industrial organizations that deploy those technologies across the world are responsible, are transparent, and are trustworthy. Of course, PR, public relations, is very important. There's a lot of people concerned about GM and GMOs and skepticism, anti-science bias, anti-vaccines, anti-development, anti-industrialization, anti-science, anti-scientist, anti-academia is very important. And we have to engage with the public. And we have to explain using key stakeholders how trustworthy technologies are, how trustworthy scientists are more often than not, and be transparent with responsible research and innovation, because why we're doing it, the responsibility and the stewardship of bringing technologies to solve the world's problems, and the magnitude of the benefit to save lives, feed the world, and breed a more sustainable agricultural system is very important. It's not me saying it as a scientist. It's patients saying how this saves and increases their lives. It's the families of patients that have genetic disease and or cancer that says this is saving their lives. Of course, it's farmers and consumers saying it's helping them save the world and then make things more efficient, make things more affordable, and make things more sustainable. And of course, we also have to entrust regulators with regulatory responsibilities. How we regulate medicines, how we regulate foods, how we regulate biotech products, how we regulate the forest, how we regulate animals and livestock, how we regulate crops is very important. And we have to systematically measure what the risks are or may be perceived or real, and what the benefits may be. Sometimes it's saving lives and curing disease. Sometimes it's more sustainable agriculture. Sometimes it's global warming and climate change. And we have to ensure, with Health Canada, for instance, for Canadians, and the NIH, and the FDA, and the USDA, and the EPA in the US, that we are being safe and efficacious and responsible. And of course, we have to do so in a pretty complex, international, interwoven, global supply chain with competitiveness at the national, at the economic, and the political level. And this is why we're here. This is why I hope some of you will pursue a scientific career, right? You can be a scientist in industry, do fundamental research, work at the university, train people, educate people, do fundamental research. You can translate sometimes that science into technology, be like an engineer, be a translational scientist, bring those technologies to the real world, and or apply that to develop products. We have to work with government. You can be a politician, a regulator, you can be a patent office, an inventor. You can be a communications expert. You can write reports. You can write public opinions. You can debate about this. You can be an industry, be an entrepreneur, an investor, or a partner, or commercializer, or a manufacturer, or a distributor. And then, of course, in society, we have to engage with the public, and we are part of the public. We have to be consumers. Hopefully, we won't have to be patients, but if we are, we'll be there in a patient's capacity. Of course, we have to deal with the media, and of course, we have to deal with ethics. And this is why, regardless of your path in your career, whether you're going to be a scientist or not, an academic or not, work in industry or not, work in government or not, hopefully be an active member of society, in the spirit of the Gairdner Foundation, we have to convene people to have those conversations. And for a reality check, no matter what your age is today, as a reminder of how hard it is to do that and how long it takes to translate science into technology and products, be reminded or note that on average, it takes 17 years for clinical evidence to become clinical practice. This means that it takes 17 years for clinical studies that are being carried out right now to become mainstream at the average practitioner. It means that a CRISPR drug being tested and successful today in 2023 in the clinic may not be widespread through the clinician system until 2040. This is what people call like lost in translation, the translational gap or the translational chasm. And we need to be more efficient and expeditious and use better science and technology to bring the very best science that we have today to patients, not in 2040, but tomorrow. And this is why I want to compel you to think about the role you may play in this community, right? The point of the Gairdner Foundation is to celebrate scientific excellence, to convene the brightest scientific minds and, of course, stakeholders, and inspire the next generation of scientists. And I hope that some of you will want to turn to researchers to invent new technologies. We'll turn into teachers, like the fantastic teachers you have in the classroom today exposing you to this, to develop and train talent. We'll turn into great speakers and ambassadors to talk about science and technology, maybe become great entrepreneurs and business people. Also went to business school, for example, founded 5 different CRISPR companies to bring those technologies to the real world. Or just learn about science and learn about technology and help spread the gospel of science. Coach people to be better scientists, better citizens, coach patients on how to navigate that, coach your family members how to accept science or not. Maybe write books about it, write papers about it, write reports about it, write stories about it. And then, of course, doing the work to administrate that and bring this to the real world, right? And I want to remind people that science is solving problems, not creating them. And whether it's Nobel Prizes in chemistry or vaccines, whether it's tackling down the pandemic, whether it's addressing an infectious disease, science is part of the solution and not part of the problem. And we are changing the world one application at a time, one patient at a time, one product at a time, one enterprise at a time, one technology at a time, to make better medicines, to make better foods, to make more sustainable forestry, and to bring the best technology to fruition. So I'll stop now. I'm saving plenty of time for a colorful Q&A. I hope you had a chance to take your notes and maybe write down your questions. And whether you're going to talk about the science, whether you're going to talk about the technology, whether you're going to talk about the products, whether we're going to talk about regulations and acceptance in either medicine, food, agriculture, biotech, forestry, or we talk about legal aspects of it and patents and lawsuits and commercialization and investments. I hope this is thought-provoking enough to trigger a good conversation, and I hope you learn to understand how important CRISPR is and how disruptive explorative CRISPR-based genome editing technologies will do in food science, in medicine, and agriculture to change the world.
Just that, just very low key, just that. I love it. It was fantastic. I feel like I've heard of CRISPR so many times, but never that angle. Like it's usually like very technical or like science. And I feel like here it's like the science is like leveraged in so many fields that are much needed, but we never discuss them. So I thought it was like a very good insight on that. The science is central, obviously, but science on its own in a lab, well, cool, but where does it, it doesn't go anywhere. So super cool, and it was funny to see like the like 2016 awards. I'm like, I think I know some faces, and then I realized like, yes, Nobel Prizes, sure, like like I've seen those faces. Yes, here they are. I feel like it's a bit less public. I don't know if the youth that are in the audience today have heard of the Gairdner Prizes before, like foundation. Like I feel like I've heard of it myself, being a scientist, like so involved in the research. Like it's a bit less publicized, like on TV than the Nobels, but there are bridges for sure. As you just showed, and then Anthony Fauci as well. Fauci?
Tony Fauci, yeah.
That I'm like, I know that face. And then it took me a while, like, no, we've seen that face so much, so many times, all of COVID. So definitely very important guys over there doing great work. I received tons of questions. So we're going to try to go through a bunch of those. And like, feel free to, jump on, elaborate a bit more on the questions itself, because I feel like you know so much and you definitely have a very broad perspective. So like, feel free to expand on the questions. So the first question was, how is AI affecting your work?
Oh, AI is a very enabling technology. So it makes the computational part of understanding genetic disease, designing those therapies, scaling up those therapies, even manufacturing those therapies, even in the context of supply chain, how to like operate and manage, different ingredients that have to come in at different times to different places to be put together and formulated. AI is systematically changing how we do the science. Fundamentally, that's mostly genetics. AI applied to genetics to find the target sequences of interest. There's AI applied to the technology to synthesize the best guides, the best LNPs, the best RNPs, the best small ingredients that go into formulating a drug. And AI goes also into manufacturing and distributing and supply chain. So AI is one of those amazingly convenient, disruptive, revolutionary technologies that is now embedded in almost everything that we do scientifically, including in genetics and manufacturing and engineering, to decipher a genetic disease and to manufacture drugs and to engineer CRISPR. So AI, AI is not new, as a reminder, right? Like when I was a student a long time ago, decades ago, literally, I did take classes on artificial intelligence and neural networks. But new modalities of AI, deep learning, machine learning with increased computational power is a game changer. And the scale and speed at which we can do that and the affordability of doing that. We don't need a computer to do that anymore. Like some average computers with average computing power can do AI and ML and DL like this right now. So we're at a time where it's exploding. It's not new per se, but it's exploding, much more scalable, much more affordable, and it's very enabling and disrupting how we do it. What's good about it is that it makes AI universally accessible to some extent. It's not just, generative AI and writing things for you and writing your essays and writing your homework and solving your issues. It's writing DNA. It's analyzing complex genetic data sets. It's, again, manufacturing new drugs more efficiently, more affordably, more cheaply, and more scalably, and very, very enabling, very, very useful, but it's just a tool.
I think it's actually like a good segue into the question that we got about how long it could take to deploy CRISPR. So I think like you touched like the 17 years between the trial and actually getting out there. But like any insights on like when do you think it could be like broadly available? Like I could go to the hospital and get it. Like
yeah.
How do you see that coming? It's a question.
So let's just say that the technology was first pioneered and developed in 2012 for genome editing. It took collectively the whole CRISPR community. It took us about 8 years to dose the first patient, Victoria Gray. 8 years for patient number 1. And then it took us about 2 years to dose 10 patients. And then just this year, we got to dose 100 patients. And I think next year we'll get to 1,000. So you can see, not quite hockey stick, but almost. You can see the pace of development and deployment of the technology is on the rise. And I think that, let's say by 2030, we'll dose north of 100,000 patients. 100,000. It's a lot. It's only 100,000, and maybe it'll be 1 million people. So I'm excited. It's very ambitious. It's very real. And we can save, maybe 1 million lives, improve 1 million lives. Now, CRISPR in crops, we're just starting right now, but it's much faster, much easier, much cheaper to deploy CRISPR in crops. And the World Economic Forum is estimating that by 2030, 100 million farms will have planted CRISPR-edited crops.
That's like tomorrow almost.
And I think, more than 1 billion people will have consumed CRISPR-edited foods. And of course, right now, we're also using CRISPR in trees. I didn't discuss that much at all, for more sustainable forestry, more efficient forestry, right? Less cumbersome forest operation, and more efficient pulping and wood manufacturing and paper manufacturing and biofuels and the like. And some of those trees we're planting now or soon will last hopefully longer than all of us. And it means that what we're doing right now in time will impact north of 10 billion people, 8 billion people that live here today, plus the next 2 billion that'll be born in the foreseeable future. So speed and scale are all relative, right? How to dose patients will take some time because we need regulators to approve this in clinical trials so we can go to phase 1, phase 2, phase 3, and then commercial stages. And I think by 2026, 27, we'll be there. Again, dosing thousands of patients in the meantime, and eventually hundreds of thousands and millions. But on the food side, it's going to be much more immediate and much larger and very visible. On the forestry side, it may not be perceived, but it'll have a fundamental impact, but it's going to take time for some of those trees to grow. So how you measure it, how you define success, and speed and scale are very important, with, of course, the understanding that the cost per person of developing one drug versus planting one tree versus planting one stalk of corn is very variable. So how we manage our resources, how we prioritize different segments, and how we elect at the individual, the national, and the corporate level to place investments and bets to advance the science and technology and deploy it for food versus ag, versus farming, versus forestry, versus biotech, versus therapeutics. There's a lot of mixes and matches to do, and we have to be strategic. There's an economic impact to it, there's a life impact to it, there's a practical impact to it.
Maybe jumping in to expand on the few things that you just said. So one on why would be more visible in crops, and then something like maybe 6 years or 8 years that you just said for the first patient. Looks like a lot, but maybe we should remind people in the audience that for science, it's actually quite fast. I'm not an expert in clinical trials or whatever. My studies are in chemistry, but still, stuff that was developed 20 years in chemical labs, or even older than that, are now starting to be in the industry. But 6-8 years for a medical trial, for me, it's amazing. But I don't know what you think.
Yeah, it typically takes 20 years, 10 to 20 years, and $1 billion to develop a drug. And 80% of the time, the pipeline doesn't make it. So it's expensive, time and resource constrained, and the percent chance of success is fairly low for fairly large commitments. In the case of CRISPR, it took us just 8 years to do the first patient, which is twice as fast as usual. And it cost, maybe between a quarter and a tenth of that. It's very substantial. So it's faster and cheaper. And to do it again and again and again, it's going to be even faster and even cheaper. And what's great about CRISPR is that it's a platform that, once demonstrated to cure one disease, that you say sickle cell disease target number 1, once we show that CRISPR works for sickle cell disease, we don't have to start from scratch to show that CRISPR works for beta thalassemia.
Gotcha. That's super impressive.
So the whole point now is that the timeline is compressed. The cost is lowered, and the scale at which we can do it is enhanced. So if you look at multidimensional elements, we can go better and cheaper and faster and more scalably. And what typically takes 10 to 20 years and $1 billion will now take maybe only 5 years and 250 million. And the next time maybe only 3 years and only 100 million. And eventually, less and less and less. On the crop side, we can go faster because the speed at which and the cost at which we can do this is much easier and more accessible. So to plant, an acre of corn is a lot more accessible than dosing one patient and telling the FDA to do that. And FDA is food and drugs. So they don't just do D, drugs, they also do F, food. And the FDA and the USDA and the EPA have been catching up with the CRISPR Craze, so to speak. And they are already food products on the market. You can see CRISPR corn here in the back. I mentioned CRISPR tomatoes are ready for sale in Japan. CRISPR tomatoes are ready for sale in the UK. I did have some edited mustard greens earlier this year that are being commercialized right now that are much less bitter. So think like less bitter, more tasty fruits and vegetables. I mentioned the non-browning mushroom, right? So less oxidizable vegetables, so they stay longer and you can enjoy them for longer and the like. Of course, crop yield and of course crop resilience to pest and frost and drought and soil erosion and the like. So, and the advantage of doing crops is every year you can do a rotation. So the speed at which we can breed crops is much faster than the speed at which we can breed humans. Think of one generation and the next generation and the next generation. It's a lot easier, a lot faster, a lot less expensive to breed corn or soy or rice or wheat or whatever you want. But there's a lot fewer people doing that. And it's not as lucrative. So in terms of speed and commitment and resources and scalability, it's a little bit of a different game, but we're very good at that game. We're very fast at that game. And you can take that to the next level and think about forestry. So some of those trees take 25 years to mature. And after 25 years, you can take 2 trees and breed them together. So if you have a 50-year career, you can maybe go through 2 rounds of breeding. As a forester, it's very underwhelming, so to speak. But with CRISPR, we can go into the lab and do that within a year or 2. So in the span of 10 years, maybe we can do 5 breeding cycles, as opposed to 125 years to do 5 breeding cycles. So we can't wait until 2048, until 2148 to do 5 breeding cycles. We can wait maybe until like 2028, and that's the game changer. So in terms of speed, in terms of scale, in terms of cost, in terms of resources, it's a game changer. It's a revolution, and we're trying to catch our breath. It's going fast. Maybe for some people and patients, it looks like we're going too slow. We're going as fast as we can. We can't go too fast because we have to be safe. We have to be responsible. We have to have all the guardrails that we need to make sure that it's done and deployed responsibly and transparently and affordably and equitably and accessibly and ethically. But we're trying to go as fast as we can.
Like COVID might have slowed down a bit of the clinical trials, for example. But
yeah, when you got to shut down your lab in the midst of a pandemic, it's took a slow down. You can only do so much work on the computer, even with AI.
Impressive, yeah. At one point, the drugs have to go into humans. Like that's that's the end goal, right? To jump on what you just said earlier, like it's going to be visible in crops, but do you really see it? The corn that you just mentioned, and tomatoes, do you actually see it? So you just mentioned a different taste for some of them, but it's not visible per se. If you don't know, you go to the grocery store, you might not notice, right?
So some are visible, some are not. So let me give you some examples. If we think about yield, it's visible to the farmer, but not visible to the consumer. If we think about drought resistance, heat resistance, cold resistance, frost resistance, pest resistance, insect resistance, yeast resistance, virus resistance, all those things are invisible to the consumer, but it's going to be more sustainable and hopefully more affordable food. So there's an aspect to it that's indirectly visible that would make the argument for, but it's operational gains. However, I showed some examples in my presentation, right? We can change the color of fish meat. And for people who like sushi, gray sushi versus red sushi is much more palatable and agreeable and acceptable and desirable, right? So in this case, you actually see the color of meat is different. I mentioned the lower bitterness mustard seeds. So if you like mustard greens or greens that are sometimes bitter, you can knock out, using CRISPR, some genes involved in bitter compounds, which presumably will make you more likely to eat your vegetables, children, and be healthier and eat a larger amount of vegetables if it's not as bitter. Now, I showed an example of a purple tomato, or purple tomato in the UK here. And this is an enhanced GABA, more nutritiously desirable tomato that tastes like a heirloom tomato more than like, your average tomato, so to speak. So if you can make tomatoes taste better and healthier, you will see it, potentially the color, or you will actually taste it. That's what we did with yogurt. We made yogurt more sustainable and more efficient. So you can taste better yogurt or afford more yogurt and eat more yogurt. And yogurt is so tasty, you might do that. Same for cheese, right? I also mentioned, you may not have seen it because I went quickly through it, the hornless cows. So cow horns are very impractical from a herd management standpoint because they can gore each other, they can hurt each other. And right now, the way to do that is somewhat barbarically or inhumanly to remove or cut their horns. What we can do with CRISPR is we can go into their DNA and cut out the gene that makes horns. And we can now breed so you can see it, right? You won't taste the difference.
Yeah.
But you can, if you go to a farm, you can see cattle without horns. So they don't have to be subjected to barbaric removal or extraction or slicing and cutting, or they don't gore each other anymore, and they live longer and better lives. So there's aspects like that that are very, very visible. A former example in the agricultural world, though it was GMO at the time, was golden rice. Right, you could actually see more vitamins in variants of corn that are enriched in vitamins to address malnutrition. So there's applications like this that we can do. And then, of course, facetiously, I showed the example of the hypoallergenic cat. Right, people are working on changing the DNA of cats to remove the immune response from humans and make cats hypoallergenic. I'm not allergic to cats. I'm not a cat person per se. I like dogs better. Again, I don't want to discriminate against anyone here per se, but some people who would sense that and who would get sick or uncomfortable around cats could now maybe get into the cat market. And you can imagine that if you can do that, I showed the example with the butterflies. If you can do that, anything you can think of, you could do. So can we make avocado with smaller seeds? Can we make grapes that are seedless? So seedless grapes exist today, but we can make them, more scalably and affordably, and they would maybe taste better and be a little bit more sweet, a bit more colorful, right? So there's all kinds of applications you can think of. Anything you can think of, theoretically, could probably be done in nature. Again, I can show you some examples here with mosquitoes, show you some examples here, with human blood. Of course, right here, we have, cattle. So, but there's things we could do like humanizing pigs so they turn into human organ donors. So if you want a kidney, let's say you have kidney failure, history of kidney failure, what we could do is, instead of taking a kidney from a human, right, you only need one. So you could take a kidney donor and somebody who's histocompatible and take the kidney, or they would give you their kidney. You don't just take it. They have to give it to you surgically, right, safely and responsibly. Or sometimes people could get organs from animals that are immunocompatible, like pigs notably can be donors for some tissues in humans. What we could do with CRISPR is we could take human cells and engineer them and inject them and make them grow into a pig and the pig itself would be engineered to be non-immunogenic. And we could grow your kidney with your DNA and your cell in a pig that's hypoallergenic, and then take your kidney from that pig and put it in you, right? Like science fiction, but just science, right?
Wow.
So we're not quite there yet. Like it's not on the market for 2023. It won't be on the market in 2024. But there's a lot of colleagues, including some of my own colleagues at NC State here at the vet school, working on using and deploying CRISPR in livestock, not just to make livestock better per se, but to humanize livestock. So we have a future not so distant in which we could grow your own tissue with your own DNA. So it's very acceptable as a recipient because it's yours, and make that work. But scalability, affordability, we're not there yet, but we're working on that.
And that's just like it's beyond words. It's beyond creativity at one point. Like it's just out of this world that you just described. Like it's something that I've heard a lot in medicine, like tailored drugs and whatnot. And I was kind of like perceiving that it could be in other fields at one point, but understanding that it's actually already extensively studied in so many fields, it's really mind blowing. And it's, as you said, like it's been so quick because the technology is fast, so that helps. But still, like it's incredible of people like taking this technology and like use that in their own field already so quickly. Like it's mind blowing completely. You actually just touched on GMOs, on food, and something that someone was asking, like, if you were to actually have tomatoes and whatnot, like engineered through CRISPR, when you get that through your own body, could you actually see that somewhere? Like that's something like just goes through as like any tomatoes? Like you find traces of something?
Yeah, so oftentimes, right? Oftentimes people think of genetically modified as transgenic, right? Where in the past, people would take a gene from one species and artificially, molecularly putting into another species. So like take genes from salmon to make, tomatoes, cold resistant, frost resistant, right? Or I mentioned putting vitamin from one species into rice to address, nutritional deficiencies and vitamin deficiencies. That's transgenic. You take a gene from one organism and put into another. Most uses of CRISPR are scientifically indistinguishable from natural breeding. So think of a tomato, a heirloom tomato that is small but tasty, and another tomato that's maybe bigger and fatter but not as tasty. And to breed them together would take a long time. And to crossbreed them together, it would take us a lot of effort, a lot of offsets to naturally find one that is a bigger tomato than the heirloom tomato, but tastes like the heirloom tomato. What we can do with CRISPR is we can make that happen in the lab. Like this, faster, easier, and cheaper. And you can't tell, like it's the same natural process as breeding. Like you can't tell whether it was made in the lab or made in nature. It's the whole point of indistinguishable. It's the same outcome. That's why it's safe. That's why it's agreeable. That's why it's not regulated, right? What you get is the same exact outcome, instead of waiting for a breeding cycle or 2 or 3 to find the one in a 10,000 or one in a million that you're looking for. We can actually make it de novo right there, right? And it will taste, hopefully not the same. It will taste the same as the heirloom, right? And you won't tell the difference. Your body doesn't tell the difference. The plant doesn't tell the difference. The soil doesn't tell the difference. The farmer doesn't tell the difference, as if it was naturally made, essentially naturally equivalent. And that's why they're not regulated in the USDA, in the FDA, in the EPA, in cases in which the outcome is indistinguishable and could be bred naturally. That's the whole point. That's the whole point. It's different from transgenics. So we're not talking about taking a gene from one species, putting it into another, and making like BT corn. Or if we humanize a pig, that's transgenic. If we take your gene and put it into the pig, it's transgenic, right? And you don't want to eat that bacon. You want to take that organ and put it in you. You don't eat it, you graft it on, right? So that's why there's all possibilities out there. But more often than not, in the food supply chain, it's a natural replicating process that is equivalent and indistinguishable.
I know you make the transitions for me for the next question that I was sent. So someone was asking about the legal on the regulation side. So you just like for food, if it tastes the same, well, good to go, like no question asked. Like I mean, that makes sense, but at the time it's very surprising. Like food is so regulated that I'm surprised. But I mean, it makes sense. It's the same.
Well, I mean, we still have to talk to the EPA and the FDA and the USDA and explain to them what we did and how we did it and why we did it, and show them the data in the field, show them the products more often than not, show them the seed, sequence the genome, tell them where it came from. Like, it's cumbersome, all encompassing, and it has to be done the right way. And we want the bar to be high, right? To convince not just the regulators, but the consumers, because it has to be safe and it has to be nutritious, right? So we want the bar to be high to some extent, high enough to clear that bar, and instill confidence and trust. That's very, very important. FDA approved or not, USDA approved or not, EPA approved or not. But it takes time to educate the regulators. It takes time to educate the consumers. It takes time to educate the scientists, you know, the work of what. No, obviously, right? So we have to have the commitment and the wisdom to do that. And it's balancing of going as fast as we can, as far as we can, and then being responsible. And that's what I call, maybe a good comment to have in mind next time you think about it. It's what I call the highway of science. So how do you actually build a highway? Right. First, you have explorers going into the wild on a dirt bike, right? Off-roading. There's no road. They're off-roading by design, right? And they're going there. They're like, you know, can we get there? Is there a road? Is there a path? And they're going to fall, break a few bones. They may or may not be wearing helmets, and then they may or may not, you know, abide by speed limits and safety guidelines. And, you know, they're explorers. They're explorers. They're exploring by the seam of their pants. Literally, that's what they're doing, right? And we want them to do that. We need them to do that. And some of them will find a path somewhere that didn't exist before, and they'll blaze a trail. And then other scientists come through, and now there's a trail that we can see. And eventually, you know, the public will come through, and eventually we'll take the trees down, we'll level the road, we'll lay down some asphalt, we'll paint lanes on it, we'll put speed limits and speed bumps and signage, and we'll put some guardrails on the side. Guardrails, the regulations. Speed limits, the regulations. Now, whether people drive correctly or not, follow the rules or not, put the seat belts on or not, put the helmets on or not, you know, I'm not going to opine on that. The point I'm trying to make, though, is that we have to understand two things. We have to understand there's a delay between the time people at the very front end are blazing new trails and the time the public gets to do that. Like, we have to understand hopefully that's not 17 years in the clinic, right? Hopefully we understand there's a gap and want to make the gap short but safe. At the same time, the guardrails are there for a reason, to prevent people from killing themselves if they drive off the road. So those guardrails are the regulations. The speed limit is the regulation. But we can never, and we should never, start with the regulation. You never put the guardrail first and then build the road. There would never be a road. There would never be explorers out there. And we have to trust that explorers that are out there without guardrails, without helmets, without safety, without regulations, are sometimes going to lead the way into a better future. And that tension between those things is the challenge here. And we have now to trust the regulators to regulate and the guardrails to be there, with the understanding that eventually there will be a guardrail, or there should be a guardrail. Maybe the guardrail should be higher or taller, or, you know, closer to the road or farther from the road, or, you know, maybe before the turn where you start building it. You can't have a guardrail all the way in every road all the time, right? It's inefficient. But in the treacherous path, in the treacherous parts, we have to have a guardrail. And those are the safety, efficacy, regulatory processes we have to jump through to make sure that the road is safe, and that is going to take us somewhere that's better in the future. But that tension of the explorers by the seam of their pants, and then second generation adopters, and then car manufacturers, and then asphalt layers, and then eventually the public going on vacation or up the mountain and down the mountain and wherever they go, right? We'll have to go there. So We can't start with a guardrail, and we can't guarantee the road is safe. And we don't want the road to be safe because it's how you explore and discover new things, right? And sometimes it works, sometimes it doesn't. And if you had to work every time, we discover nothing. And if we failed every time, it'd be just a bad outcome and a waste of time. So that tension of success rate, is a challenge, but we have to accept that and tolerate that. And we have to have people who want to embrace that and do that, and disruptors. Disrupt enough, but not too much, right? If you're a revolutionary, you're challenging a lot of status quo. You can't do crazy things too much, but it's by being creative, right? And inventing new things and doing new things in different ways for different applications, different people, that eventually some of those things turn into real beneficial products that are changing the world.
I think it makes sense. That sounds about a train, then you don't really know sometimes where you're going, but you're like opening a few boxes at the time and like seeing where it lands, and you can have the regulations before even starting to know where you're gonna end. Like, who could have imagined that it could be my DNA in pigs or organ transplant a few years ago? Like, no regulations could have been put in place before someone actually even tries it. So that's a good point of like, you have to try it first. We have a few more minutes. Maybe I want to switch like a very different topics. Someone was wondering about experiences like that you've tried, like dealing in the classroom that you mentioned earlier, that high schoolers are coming, like, because it looks so complicated, as in like, it's easy for science, but it looks like very, like, a lot of materials and technologies and whatnot. So what do you do with kids actually?
So there are kits online for kids, right? To do that, in your high school, you can put a gene that will be a fluorescent protein like GFP or M-Sherry, and put that into a bacterium.
Okay.
And make it fluoresce, right? So you can actually see it. You could taste it if you really wanted to, but you shouldn't, right? So there are things you can do in basic microbiology that allows you, in a lab setting of the average high school, even sometimes the average middle school in Canada and the US for today's audience, to do some of those experiments. And then, deliver DNA with or without CRISPR to a bacterium that now becomes fluorescent.
Wow. So it's already accessible in some way. So you can't get your own drug tomorrow, but you can
kids do not make your own drug at home.
Yeah.
It's not advisable. That's why there's a pharmacology industry that does that. But if you want to try to do, GFP fluorescent E. coli in the lab in your high school, talk to your science teacher. It should be agreeable, accessible, affordable, and mind-blowing.
So does that mean that farmers and like forest, like people working in the forest industry could also do that on their own? Like, it's not necessarily like there is centralized, like there is this one big company making those seeds for everyone. It could be a bit more like you do your own.
We want to democratize that. We want to democratize breeding. So, not everybody should be a surgeon. So if I give you a scalpel, doesn't mean you're a surgeon. You need, surgery training, right? Maybe if you cut yourself, you can, sew like a little scar, but you don't want to do that at home for sure. So the scalpel does not a surgeon make. So CRISPR does not a breeder make, right? It's not that trivial. Like, I'm going to put this gene into my crop and I'm going to make wine better, or beer better, or whiskey better, or rice better, or like, it's not that trivial. Right, trust me. But somebody who's a breeder could get access to CRISPR and breed better and faster and more affordably, and do more cycles of breeding over the course of their lifespan. That's the revolutionary of the technology. Yeah.
Well, looking forward to see what's coming up in a few years, like even a few decades. Like, it seems like with all of time, we could actually see some pretty major changes. So very excited to see that. It's impressive. And again, like, thank you so much for opening horizons with us, because I definitely have heard about it, but I feel like I've heard like not even the tip of the iceberg, like a drop of the iceberg. I don't even know. Very impressive. So thank you so much for your time, on all your expertise. Very accessible and just mind-blowing. That's the key word I'm gonna leave you with. Mind-blowing, honestly. So thank you so much for the terrific. And I feel like we're gonna hear from you again for sure. I feel like you're doing such terrific work that you won't disappear. Like, we're gonna see you on the news and whatnot over the next years for sure. So thank you so much for your time. Thanks for the students that were with us today, for your great questions. I have so many more, but time is running out. So we're gonna thank Rodolphe for going back to our lives and waiting to see what CRISPR is gonna bring us, or scientists are gonna bring us in a few years then. Thank you.
Indeed. Thank you all. Cheers.
Bye.